Hello students, hello everybody. Here, this is Dr. Preeti Mathur, Associate Professor, Department of Zoology, SPC, Government College Ajmer. Here, again I have come with the same topic, that is Biofertilizers Part IV. This will be the concluding part of my lecture. Azolla as a biofertilizer. Azolla is a tiny freshwater fern common in ponds, ditches, and rice fields. It has been used as a biofertilizer for a rice in all major rice growing countries, including India, Thailand, Korea, Philippines, Brazil, and West Africa. The nitrogen fixing work is accomplished by the symbiotic relationship between the fern and BGA, that is blue-green algae, Anabina azolla. In addition to nitrogen, the decomposed azolla also provides potassium, phosphorus, zinc, and iron to the crop. Now here in this slide, I'll show you the properties of azolla. It has various qualities, like the biomass of azolla gets doubled within 5 to 7 days by vegetative methods. And this azolla, it fixes 40 to 80 kilogram nitrogen per hectare per year. Now again, this azolla, it is a good manure for flooded rice. Azolla increase of crop yield up to 15 to 20%, and it has been observed while fertilizing the rice with azolla. Hybrids are growing faster in azolla. It is also seen that it is tolerant to heat and cold. Generally, it fixes 4 to 5% more nitrogen. Now, how can we do the biofertilizer? How can we use biofertilizers application methods? There are 3 ways of using these nitrogen fixing phosphorus soluble microbes. P.S.M. Shortly I am using it, phosphorus solubilizing microbes. We call it, the short form is P.S.M. here. These are the bacterias. They help in seed treatment, root dipping, and soil applications. Seed treatment. Seed treatment is the most common method adopted for all types of inoculant. The seed treatment is effective and economic. Seed treatment with Rhizobium, Azotobacter, Azospirillum, along with P.S.M. Now next point is, the seed treatment can be done with any of 2 or more bacteria. Here there is no side effect. In seed treatment there is no side effect. And lastly, important things, as the seeds must be coated first with Rhizobium or Azotobacter or Azospirillum. When each seeds get a layer of above bacteria, then the P.S.M. inoculant has to be treated on outer layer of the seeds. This method will provide maximum number of population of each bacteria required for better results. Mixing the any of 2 bacteria and the treatment of seed will not provide maximum number of bacteria of individuals. Root dipping. Application of Azospirillum with the paddy or vegetable plants. This method is needed. The required quantity of Azospirillum has to be mixed with 5 to 10 liters of water at one corner of the field, and all plants have to kept for minimum half an hour before sowing. Soil application. P.S.M has to be used as a soil application. Use of 2 bags of P.S.M per acre. Mix P.S.M with 400 to 600 kilograms of cow dung along with half bag of rock phosphate if available, and the mixture of P.S.M, cow dung and rock phosphate have to be kept under any tree shade or ceiling for overnight and maintain 50% moisture. Use the moisture as a soil application in rows or during leveling of soil. Now, viewers, we have to be very conscious about the precautions. Store biofertilizer packets in cool and dry place away from direct sunlight and heat. Use right combination of biofertilizers. Rhizobium is crop specific, so use in specified crop. Do not mix with chemicals. And lastly, the important thing is use the packet before expiry, only on the specified crop by the recommended method. Now students will discuss about the advantages of biofertilizers. The renewable source of nutrients sustains soil health. Secondly, supplement chemical fertilizers, replace 25 to 30% chemical fertilizers, increase the grain yields by 10 to 40%. They decompose plant residues and stabilize carbon nitrogen ratio of soil. It also improves texture, structure and water holding capacity of soil. So there is no adverse effect on plant growth and soil fertility. It stimulates plant growth by secreting growth hormones. It secretes fungistatic and antibiotic-like substances, solubilize and mobilize nutrients. And finally, eco-friendly, non-pollutants and cost-effective method. Now about the disadvantages. We can very well know that the biofertilizers require a special care for long-term storage because they are alive. They must be used before their expiry date. If other microorganisms contaminate the carrier medium, or if growers use the wrong strain, they are not as effective. Biofertilizers lose their effectiveness if the soil is too hot or dry. So, this was all about biofertilizers, viewers, and my dear students. This was the 4th part of my lecture of biofertilizers, and now I conclude here, and I again thank you very much for patiently hearing me, listening me. My name is Dr. Preeti Mathur, Associate Professor, Department of Zoology, SPC, Government College, Ajmer. We'll meet in my next lecture. If you have any query, you can see my email address, that is preetimathur23@yahoo.co.in. Okay, it's written below my name. Thank you.